In this video, we're going to cover resting membrane potential, graded potentials, and action potentials of neurons. Let's start this lecture by first defining what a membrane potential is. What does this mean? Membrane potential refers to the difference in electrical charge between the inside and outside of the cell. It's the separation of opposing charges across the membrane. The greater the number of charges separated, the larger the potential. And all living cells have a membrane potential, but in this lecture, we're going to be talking about neurons. Now, this is measured in volts, millivolts. So resting membrane potential is the voltage difference across the membrane when cells are at rest. Meaning they are not producing electrical signals. The resting membrane potential of a typical neuron is approximately negative 70 millivolts because the inside of the neuron is more negatively charged than the outside. Let's break down how we generate and maintain this resting membrane potential. An average of negative 70 millivolts. The difference in electrical charge is caused by the uneven distribution of ions on the inside and outside of the membrane. Ions are atoms that have either lost or gained electrons, and so they have a positive or negative charge. A positive ion is called a cation, and a negative ion is called an anion, okay? And the human body contains a variety of ions. At rest, there is a higher concentration of sodium and chloride on the outside, and a higher concentration of potassium and negatively charged anions, represented here by A- on the inside of the cell. These anions can include amino acids, proteins, and other metabolites that have a negative charge, okay? Now, there are two major things that determine or establish the resting membrane potential. First is the differences in ion concentrations in the inside and outside of the cell, and the differences in membrane permeabilities to the different ions. With that said, we're going to be focusing on sodium and potassium ions because these are the ions that directly influence or produce the resting membrane potential. In some cells, chloride ions play a role, but let's just focus on sodium and potassium. So now let's subtract complexity and first take a look at the sodium-potassium ATPase pump, because this pump is responsible for maintaining the concentration gradient for potassium and sodium. Okay, this transport protein constantly pumps 3 sodium ions out of the cell and 2 potassium ions into the cell. It requires energy to do so. It requires ATP. Both of these ions are positive ions, and so there are more positive ions being pumped out, making the inside of the cell more negative, okay? The pumping of sodium ions out of the cell leads to a higher concentration of sodium outside the cell in comparison to the inside. And potassium concentration inside the cell is larger than it is outside due to the pumping of potassium ions into the cell. Here's the thing though, this pump only separates enough charges to provide a membrane potential of 1 to 3 millivolts, with the inside of the cell being negatively charged, okay? Its contribution is quite small because remember, the resting membrane potential is approximately negative 70 millivolts. We're only at negative 3 millivolts. Negative 3 millivolts. So where do we get this membrane potential then? Most of the membrane potential is produced by potassium and sodium passively diffusing down their concentration gradient. Okay, let's go through the movement of potassium through leaky potassium channels or leak channels. We've already established that potassium is more concentrated in the intracellular fluid inside the cell. And so there will be a strong concentration gradient moving potassium out of the cell through open potassium channels called leak channels, okay? It's going to move down its concentration gradient from high concentration to low concentration. And this is going to make the outside of the cell more positive, okay? Because potassium is carrying its positivity out of the cell. Now, remember earlier we mentioned how there are negatively charged ions or anions inside the cell. The plasma membrane is impermeable to these large anions. They aren't able to go through the leak channels, even though they have a significant concentration gradient. This door just isn't meant for them, okay? And so potassium leaves behind unpaired anions. As a result, the inside of the cell is more negative. What happens then? As time goes on, as more potassium ions leave the cell and the inside of the cell becomes more negative, an electrical gradient is generated. Potassium would be drawn to the negatively charged interior and drawn away from the positively charged exterior. So now we have two opposing forces. The concentration gradient generally pushing potassium out of the cell, and the electrical gradient moving potassium back into the cell. At the start, the concentration gradient will be greater than the electrical gradient, so there's going to be a net movement of potassium out of the cell. But over time, the movement of potassium out of the cell due to the concentration gradient would equal the movement of potassium back into the cell, as the electrical gradient would also become stronger. So the movement of potassium outside the cell would eventually equal the movement of potassium into the cell. This is known as the equilibrium potential for potassium. Potassium will bring the membrane potential to negative 90 millivolts. Here's the thing though. This is true if we're only dealing with a single ion. If we're only dealing with potassium, the equilibrium potential equals the resting membrane potential. But as you'll see, the resting membrane potential will change as we're dealing with multiple ions. Now, this 90 millivolts is the potential, the electrical potential, for driving potassium into the cell to balance the concentration gradient that is pushing potassium out of the cell. And the negative sign is used to indicate the polarity of the extra charge inside the membrane. So if this was positive 90 millivolts, it's the same magnitude, but it just means that the inside would be more positive than the outside. With that said, the equilibrium potential for an ion depends on two things. I think I mentioned it earlier. The concentration gradient for the ion across the membrane and the cell being permeable to that ion. So the greater the concentration gradient, the greater the equilibrium potential, because a larger movement of ions, electrically driven, will be needed to balance the concentration difference movement. All right? That's the potassium movement. Let's move on to sodium. Similar situation for sodium. We've established that the concentration of sodium is higher outside the cell, and so the concentration gradient for sodium would move these ions into the cell, making the inside of the cell more positive. On the outside, there are chloride ions, okay? These blue ones here, because sodium and chloride are the key ions found in the extracellular space. As sodium moves into the cell, chloride ions are left behind, making the outside more negative. Same thing as potassium, there's also going to be an electrical gradient that's going to move sodium out of the cell, and the net movement of sodium into the cell will continue until the outward electrical gradient and the inward concentration gradient balances each other out. That is, the concentration gradient is equal but opposite to the movement due to the electrical gradient. And the equilibrium potential for sodium is at positive 60 millivolts. Okay, but here's the thing though. The membrane is way more permeable to potassium than it is to sodium, which means large amounts of potassium will move out of the cell, making the cell more negative. But at the same time, the membrane, this neuron, allows a small amount of sodium into the cell, bringing the resting potential to negative 70 millivolts, which is less than the equilibrium potential for potassium, which is negative 90 millivolts, okay? And this leads us to the Nernst equation, which is the formula that tells us the equilibrium potential for each ion. This is how we can calculate the equilibrium potential for any ion. So this equilibrium potential for a particular ion in millivolts equals 61. This 61 here is a constant, and it's divided by the valence of the ion. So for potassium and sodium, this is 1. They both have a charge of +1, times the log of the concentration of the ion outside the cell divided by the concentration of the ion inside the cell. And the concentration is given to you in a table or something, so you can just plug in the values into the formula and you get your answer. Okay, so based on how much of these ions is moving across the membrane at any given time, the actual resting membrane potential of the cell will ultimately be somewhere in between all of these different membrane potentials. In this case, the membrane is way more permeable to potassium, let's say 90%, so that we can take these proportions and multiply them by the equilibrium potential and then add up the total to get the resting membrane potential of the cell. This gives us a total of around -70 millivolts. Okay, that's the resting membrane potential. Let's do a quick summary before we move on. So resting membrane potential is the difference in electrical charge between the inside and outside of a neuron at rest. An ion's equilibrium potential is the point where its concentration gradient equals its electrical gradient, and we can calculate this using the Nernst equation. Therefore, based on each ion's relative permeability, the resting membrane potential of the cell equals the total of the equilibrium potentials of each individual ion. That's resting membrane potential. Let's move on to graded potentials. Okay, we've covered that the resting membrane potential is when a cell is electrically at rest, and the resting membrane of a typical neuron is -70 millivolts. And we know neurons use electrical signals to receive, process, and transmit messages. Here's the thing. A nerve impulse, or an action potential, is only initiated if a threshold potential is reached. This is typically between -50 and -55 millivolts. When a neuron reaches this point, it will fire an action potential. Before we break down graded potentials, let's first go through several terms that are used to describe changes in potential. First is polarization. This is when the membrane potential is other than 0 millivolts in either the positive or negative direction. There's a voltage difference across the membrane. Remember that the sign of the potential, plus or minus, shows whether there are excess positive or excess negative charges on the inside of the membrane. Next is depolarization. The membrane is depolarized when its potential becomes less negative. It's closer to 0 millivolts. So for example, from -70 millivolts to -60 millivolts, okay? Then there's repolarization. The membrane potential that has been depolarized returns to its resting potential. And lastly, there's hyperpolarization. The membrane is hyperpolarized when the potential is more negative than the resting potential. It's farther from 0 millivolts. So for example, from -70 millivolts to -80 millivolts. Hyper meaning more negative. The inside becomes more negative. A graded potential then is a small change from the resting membrane potential that makes the membrane either more polarized or less polarized, making the inside of the cell more negative or less negative. And there are different types of graded potentials: receptor potentials, end plate potentials, pacemaker potentials. But what we're looking at are postsynaptic potentials in neurons. Okay? Let's break this down further. Recall from the neuron anatomy lecture, neurons communicate with each other via a junction called a synapse. This is a presynaptic neuron, well, a part of a presynaptic neuron, and the postsynaptic neuron, or postsynaptic membrane. A neuron receives a variety of inputs from presynaptic neurons. We have two showing here in blue and red. Presynaptic neurons are those that conduct signals towards synapses, and postsynaptic neurons are those that conduct signals away from synapses. Most synapses relay signals from one neuron to another via chemical messengers called neurotransmitters. Each presynaptic neuron normally produces only one neurotransmitter. However, the neurotransmitter released by different neurons varies, okay? And there are two types of synapses based on the ion permeability changes that occur. We have excitatory synapses and inhibitory synapses. Let's go through what happens with this excitatory presynaptic neuron, this blue one here. Okay, I've simplified this drawing here on the top left to make it more easier to go through the overview picture. Starting with excitatory, this neuron releases excitatory neurotransmitters. An example is glutamate. Glutamate will bind to receptors on this postsynaptic membrane, and this binding is going to cause ligand-gated ion channels to open. Ligand-gated channels change shape in response to the binding of a specific molecule. In this case, neurotransmitters. We have glutamate. It's either a sodium or calcium channel, but for this case, it's sodium. So sodium will flow into the cell, and the movement of sodium into the cell depolarizes the membrane, producing a graded potential. The inside of the membrane is slightly less negative than at resting potential. This slight depolarization is bringing the postsynaptic neurons membrane closer to threshold. And this change is called an excitatory postsynaptic potential, or EPSP. It's bringing this postsynaptic neuron closer to threshold potential, to its firing threshold. Now, a single EPSP causes only a small change on the membrane potential. So to reach the threshold potential, a large number of these inputs are required. You'll see what this means in a minute. Moving on to inhibitory neurotransmitters, this inhibitory red synapse. An example is the neurotransmitter GABA, gamma-aminobutyric acid. Inhibitory neurotransmitters causes hyperpolarization, a small hyperpolarization. When GABA binds to this receptor, it's going to allow the movement of potassium ions out of the cell and chloride ions into the cell, which results in a net gain of negative charge. It's leaving more negative charges behind. Recall that as potassium ions are moving out of the cell, they are leaving behind anions, making the inside of the cell more negative. This is going to cause the membrane potential to move farther from zero, farther from threshold, way far. And this is referred to as an inhibitory postsynaptic potential, IPSP. Okay? EPSPs and IPSPs are graded potentials. So, taking all of this into account, the combined effect of all neurotransmitters acting on a target neuron determines whether a threshold potential is achieved. This is referred to as the grand postsynaptic potential, GPSP, which represents the entire potential in the postsynaptic neuron. It's made up of all concurrent EPSPs and IPSPs, okay? Now, if the threshold potential that's required is around negative 55 millivolts, how do we reach this threshold? Great question. An action potential occurs, meaning that a neuron will fire if the combination of graded potentials exceeds a threshold. The postsynaptic neuron can achieve threshold by either temporal summation or spatial summation. Summation is the combination or addition of these impulses. Let's go through this. Let's first go through no summation. So let's say this blue excitatory presynaptic neuron has an action potential that causes an EPSP in the postsynaptic neuron. After the EPSP has died off, there's another action potential in the presynaptic neuron. A second EPSP of the same magnitude will happen. There's no summation here because the first EPSP has died off. So moving on to temporal summation. Temporal summation involves one presynaptic neuron. If this presynaptic neuron is triggered a second time before the first EPSP has died off, then the second EPSP will add on to the first EPSP, resulting in temporal summation. And if enough EPSPs are happening very close together, okay, before the previous EPSP has died off, the threshold can be achieved. This is known as temporal summation. Think of temporal summation as a one-man show, okay? So this single presynaptic neuron is like, hey guys, relax, I've got this. I'm going to reach that threshold and fire an action potential by myself. Watch me, okay? And it's going to cause an EPSP in the postsynaptic neuron before the previous EPSP has died off, okay? Spatial summation, on the other hand, think about it as teamwork. There are multiple presynaptic inputs, spatially separated neurons that are stimulated at the same time, and they are generating an EPSP in the postsynaptic neuron. So these multiple presynaptic inputs include this blue, green, and purple inputs. And the addition of these EPSPs increases the chance of reaching threshold to initiate an action potential. That's EPSPs. Now, this also applies to IPSPs, inhibitory. It can undergo both temporal and spatial summation. So if we add IPSPs together, it's going to move the potential farther from threshold, okay? Now, another thing as well, if an excitatory and inhibitory signals are happening at the same time, it cancels each other out. It doesn't change the postsynaptic potential, it remains close to resting potential, okay? It's like they're fighting each other, yeah, we're just gonna cancel each other out. So in this example, the postsynaptic neuron may be receiving excitatory signals from glutamate and inhibitory signals from GABA. and they're just canceling each other out. So in order to fire an action potential, we want more excitatory inputs. We want more of the positive inputs, okay? That's graded potentials. Let's move on to action potentials. This is a beauty. Action potentials down the axon are the method the nervous system uses to communicate from cell to cell. So if we take a look at the structure of a neuron, okay, look at this masterpiece. It typically consists of the cell body, the nucleus, the dendrites, and the axon. The dendrites carry signals toward the cell body, and these two structures are what's known as the neuron's input zone. These are the part of a neuron that receive and integrate incoming signals from other neurons. Graded potentials are generated in this area in response to a stimuli, in this case, incoming chemical messengers. And then there's the axon hillock, which is this site where action potentials are initiated by a summation of graded potentials when the threshold is achieved. The action potential, the nerve impulse, is then carried along the axon from the axon hillock, which is also referred to as the trigger zone, all the way to the axon terminals, which are these branched endings here. So we can identify the axon as the conduction zone. And then these terminals release chemical messages, just like we covered in graded potentials. There's presynaptic neurons which act on other cells, okay? And we can label this part as the output zone. So we have the input zone where incoming signals are received and processed by the dendrites and cell body. The trigger zone, the axon hillock, this is where the action potential is initiated. Then the conduction zone and output zone. So let's take everything we've covered and break down the components involved during an action potential. The resting membrane potential is at -70 millivolts, and we covered how a single EPSP causes only a small change in the membrane potential. But if we added all the excitatory impulses and it brings the resting membrane potential to a specific threshold value of about -55 millivolts, this is going to trigger an action potential. And this is where the voltage-gated ion channels come in. In the graded potentials, we talked about ligand-gated channels that open in response to the binding of a specific molecule, in this case, a neurotransmitter. With action potentials, this is where voltage-gated ion channels come in. These channels are what allow action potentials to occur. There are two types of channels that we're interested in: voltage-gated sodium channels and voltage-gated potassium channels. Voltage-gated sodium channels have two gates, an activation gate, okay, and an inactivation gate. The activation gate is sort of like a sliding door that opens when it's activated. And the inactivation gate, well, we can describe this as a ball and chain-like structure, okay? And this gate is open when the ball is just chilling free at the end of its chain, and it is closed when the ball binds to the channel opening, blocking the opening. In order for sodium ions to pass through, both gates must be open, because if either gate is closed, well, sodium ions can't pass through. So we can say that the voltage-gated sodium channels have three conformations, three states. It can be closed, but capable of opening. The inactivation gate is open because the ball is just hanging free. It can be completely open and activated, so both gates are open, okay? Or it can be closed and not capable of opening. It's inactivated. This is the inactivation state. And voltage-gated sodium channels move through these conformations as a result of voltage changes during an action potential, which we'll break down. When it comes to voltage-gated potassium channels, they only have an activation gate, this right here, which can either be open or closed, okay? Now that we've established these two channels, let's go through what happens during an action potential. At resting potential, all voltage-gated channels are closed. The activation gate is closed, but the inactivation gate is open. It's in the state of being closed, but capable of opening. Then, at threshold potential, we've triggered an action potential. The membrane potential is at -55 millivolts. So think about all the added graded potentials. Once an action potential is triggered at the axon hillock, we don't require any other stimuli to activate the remaining nerve fiber, okay? So just focusing on the axon hillock here, let's work our way down the axon and explain what happens. This part here is called the active area, okay? The remainder of this axon is still at resting potential, the inactive areas. These negative signs indicate that. What happens is sodium channels in this area open and are activated, meaning the activation gate here is open and the inactivation gate is already open. And sodium ions are going to flow into the cell. The gates have opened and they're flooding in, causing massive depolarization. Remember what depolarization means. It's making the inside of the cell less negative. And the potential is going to spike to positive 30 millivolts. This is the rising phase of an action potential. The flow or influx of sodium ions causes nearby voltage-gated sodium channels to open up as well. And this is going to set off a chain of reactions that continue down the entire length of the axon. This is our action potential. The neuron has fired. It's like bang, 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 let's go. The gates of sodiums have opened. Okay, so the nearby inactive areas are depolarized because of the flow of sodium ions. We'll come back to this and you'll see what I mean. Focusing back on the axon hillock area, as soon as we've hit the peak of action potential, the sodium inactivation gate closes and sodium ions can't enter anymore, okay? Sodium permeability decreases. At the same time that the sodium channels are inactivated, the voltage-gated potassium channels start to open at the peak of the action potential. This is a delayed response though, okay? Now, why are the potassium channels opening? Because we need to return the membrane potential to its resting potential now. By opening the potassium channels, potassium ions are going to flow out of the cell, down its electrochemical gradient. As a result, it's going to gradually restore the membrane potential. This is the falling phase of an action potential. And as the membrane potential returns to its resting potential, the voltage sodium channels change to their closed but capable of opening conformation. As we return to resting potential, remember the potassium leak channels, okay? The potassium leak channels are going to do their job and potassium will continue to leak out. However, because these voltage-gated potassium channels are slow to close, more potassium ions may leave, making the inside of the cell even more negative than its resting potential, briefly hyperpolarizing the membrane to negative 90 millivolts, okay? Eventually, the voltage-gated potassium channels all close and the membrane returns to resting potential until another triggering event happens, okay? Now, in regards to the remainder of this axon, let's go through this. As sodium ions are flowing in, these ions will depolarize the adjacent inactive area from resting potential to threshold potential, from negative 70 to negative 55 millivolts. And the previous active area is returned to resting potential. And everything we broke down will happen to this active area and down the axon. The cycle repeats itself down the length of the axon. And the previous active areas are in their refractory period, meaning a new action potential can't be triggered in an area that has just undergone an action potential. This is where the terms absolute and relative refractory period comes in. The difference is that the relative refractory period is when the cell is hyperpolarized. It's difficult to fire an action potential. It would require a greater triggering event to fire another action potential. An absolute refractory period is a time frame in which a neuron just can't initiate another action potential. This occurs after an action potential. Once the voltage-gated sodium channels open, they can't open again in response to another triggering event, even if the stimuli is strong, until they reset to their closed and capable of opening conformation when resting potential is restored. Okay? Let's take everything and summarize this in a graph before moving forward with what happens after an action potential reaches the axon terminals. Okay, at resting potential, all voltage-gated channels are closed. Then, once the threshold has been reached, the sodium channels open, and sodium flows into the cell, causing depolarization to positive 30 millivolts, the rising phase of an action potential, because sodium ions are flowing into the cell. During the repolarization phase, the sodium inactivation gate closes and sodium permeability decreases, while the potassium activation gate opens and potassium leaves the cell, bringing the membrane potential to its resting potential. This is the falling phase, okay? As potassium leaves the cell, the membrane is hyperpolarized, okay, making it even more negative, and eventually the potassium activation gate closes and the membrane returns to its resting potential. That's action potential. Absolutely gorgeous process. Let's now go through what happens when an action potential reaches the axon terminal. This is going to trigger the opening of voltage-gated calcium channels. This right here in the synaptic knob. And calcium is going to flow into the synaptic knob. And what calcium does is It stimulates the release of neurotransmitters from these synaptic vesicles into the synaptic cleft, and this occurs via exercitosis. Then these neurotransmitters diffuse across and bind with specific protein receptors on the postsynaptic membrane, and their activity is going to influence the activity of this postsynaptic neuron. And like we covered in the graded potential part of this lecture, each presynaptic neuron generally only releases one neurotransmitter, which causes different ion permeability changes. And synapses can be excitatory or inhibitory. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video, or you can view the entire playlist. Hey, stop procrastinating!